Research Unit, and we sponsor this event and have been doing so for the last couple of years. Great to have such a wonderful turnout. And it is my pleasure today to introduce Soren Brothers, not the Soren Brothers, but just Soren Brothers, who is our replacement for our limnologist Wayne Wurzbach, who many of you know because he's been working with us for hundreds of years. Soren is a limnologist from Toronto, Ontario. He's very, very diverse, so we're one of the reasons why we're excited to have him. He's got a bachelor's in conservation biology with a minor in political science from the University of British Columbia in Vancouver. He did his master's in Montreal, Quebec, where he examined the effects of landscape flooding by boreal hydroelectric reservoirs. And he did his PhD in Berlin, Germany at a top-notch Institute of Freshwater Ecology and Inland Fisheries at Potsdam University. And there, he looked at the effects of regime shifts between turbid and clear water states on the primary production and carbon cycling of shallow lakes, which is exactly what we have happening here in Utah Utah Lake. So he's a good fit for that. He talks funny. We've been working on him. He's from Canada. He's had a little coaching on some of his words, but besides that, we're very excited to have him because he's got incredible depth and he also has incredible breadth, as you'll see today. And he's super excited to work in Utah's lakes and reservoirs. And he's a sound enough scientist and he's diverse enough that we might even be able to convince him to work in a large river someday. The next brown bag seminar, we decided we shouldn't call them brown bag because that suggests you should bring your own food. But our next science seminar is November 2nd, same time, same place. And it's Michael Cole, who's with Terry Messmer, and they're going to be talking about sage grouse. Sorry.
Hey, thanks so much, Freda. Yeah, so I've just been here a couple months. I arrived around the end of July, and I'm mostly giving this talk as a way to make friends. And, I need to meet local people. So here I am. But also, I just thought I'd be able to introduce a few ideas of limnology or a few aspects of what I've been working on that I thought might be useful in terms of management or restoration concerns in Utah right now, and just having a bit of a discourse and seeing what you guys are interested in from me. So, yeah. So I talking about a changing world in this title. I don't know if any of you, or how many of you read the abstract, but to give a bit of an idea, I took a picture of, this is from the top of the Uintas, looking down at some lakes there. And something that looks like a really beautiful scene. But the question is, in terms of a changing world is, how have these lakes been changing, or how are they changing? Are these lakes in their natural state, or, for example, maybe one of these lakes is a reservoir, you can tell from the area on the sides, and how does that influence the natural community of the lake? Are there invasive species in these lakes? So a lot of the high Uintas lakes, mountains, traditionally didn't have fish, but then they were stocked with fish early on, and still maybe now. Are the nutrient levels of these lakes at historic levels? So, for example, usually that's an issue coming from Ontario. We think of just associated with agriculture and eutrophication of lower elevation lakes, but I know Dr. Janice Brainy at USU, for example, is working with dust deposition of nutrients in these high mountain lakes with more droughts. So nutrients could even be changing. You also have changing temperature and precipitation patterns in these lakes. So basically what looks to us like a beautiful set of lakes high in the mountains, totally cut off from humanity, isn't necessarily that case. And I'm really interested in looking at these shifts in new patterns and old ones. So as I mentioned, I'm a newcomer here. I'd never really been to Utah before a couple months ago. So this is something where I'd want a bit more feedback from people at the end. But these are what I identified so far as what seems to me to be the most important issues in Utah. One is water quality. Wayne told me not to use this picture because he said everyone's really tired of it here. But that's his hand showing eutrophication of Farmington Bay, Great Salt Lake. Fish, I've heard, are pretty popular in Utah. I don't know what kind of fish that is. Maybe someone can tell me later, too. Yeah, I'm just kind of joking. I know it's a And then water quantity seems to be an issue. So. But yeah, so briefly what I study. I tried just doing a Google search of basic food web, and this is the most basic food web image I could get for a lake, or basic kind of lake cross-section. You have nutrients going in, you have zooplankton, pelagic fish, feeding, kind of cycling through with the birds. You have benthic algae here. and macrophytes, so large plants underwater. And that's mostly where I'll be focusing on, and looking, most of the history of limnology has focused on these guys, phytoplankton, and what's happening out in the open water. And a lot of my work has been really a lot more near shore, and it's trying to integrate that, not saying that that's the only thing that matters, but just integrating it better with our understanding of how the whole ecosystem works. And a few sort of MVPs, most valuable players. It's a bit of an introduction just for some of the words I'll be using if they're not familiar. So algae, most people know, but there's phytoplankton, which is those offshore, the algae actually growing in the water column. And then what I've been working on more is periphyton, which is the benthic community. So I'm especially thinking of the algae that's growing on the surfaces. So this is algae growing on a piece of plastic I put in a lake in Germany. There's also macrophytes, so those are the large plants that are growing underwater. And they can be also considered plants that are coming out of the water, like lily pads and reeds. And there's cyanobacteria. So all of these things, primary producers, are just whatever are taking carbon dioxide, converting it to food, releasing oxygen into the lake. So the question is, why do we care? Why have I spent so much time on this? A big part is food supply. So as I mentioned, primary producers in lakes, they are creating food. They're turning carbon dioxide into organic carbon, which animals eat. So this is the base of the food web within the lakes. So if you're able to quantify the primary production in the lake, you're getting an idea of actually what the capacity is for that ecosystem to support higher levels of the food web up to fish. It's also oxygen supply into the ecosystem. So what's happening with the algal production can have really strong influences, either supplying lots of oxygen to fish and macroinvertebrates and whatever else is in the aquatic ecosystem. And it can also be involved, as I'm going to show later, in terms of taking that away and producing these hypoxic or anoxic conditions where you can have huge fish kills. And something I've been working on, I'm going to just go over really quickly later on. That's something that's interested me a lot, looking at carbon cycling, and especially climate change. So lakes pump a lot of carbon dioxide, tend to pump carbon dioxide into the atmosphere, and I find that feedback and the role of aquatic systems as a part of the regulation of the Earth's atmospheric CO2 concentrations to be really interesting. And that's maybe not, a direct funding exciting issue here, but it's something I think is kind of cool. So I'm going to go over that a little bit too. And then, so kind of tying in with oxygen supply, primary production is also important just for the water quality and looking at harmful algal blooms, and they can actually, you can have a lot of these interactions between the different groups. So I'm going to try and argue that benthic primary production is really important, and I can use a lot of pretty recent work that's been showing to support that. So this, I realize none of you can actually read this, so it says mean benthic contribution to fish body carbon. So that's a measure of actually what the fish are eating from what has grown at the sediment surface. So not just what's settled down there, but looking at, for example, so the average here is 57%. This is from a study from 2011. And so this means that about 57% of all these lakes that they studied, the average, so the average fish was made up of 57% of the benthic zone. That's the mean of what the benthic contribution was to fish. So it's more than half in most of these studies. And so that means the remainder would be whatever was in the water column, so looking at that phytoplankton. And then this is benthic contribution to whole lake primary production. So this is saying, and then across mean depth. So the deeper a lake you have, you expect there to be less benthic contribution to the lake, because benthic primary production is usually light limited. So the deeper the lake it is, the less light you're having going to the sediments, and you're going to have less growing down there. Well, you can see, again, this goes up to 100%. And this is a log scale, so it's 1, 10, 100, and 1,000 meters deep. And so just out of curiosity, I put Utah Lake, Great Salt Lake, Bear Lake, and Flaming Gorge. So don't mind the height of those red things. Those are just looking at where they are in terms of the mean depths of those lakes. And so you can see that in those lakes you could have depending on the turbidity of the system and these other factors in different times of the year, you can have almost 100% of the production in those lakes theoretically being benthic. So they are important for these lakes. And this is a graph from Vadaboncoeur et al. 2002. She's over at Wright State University in Ohio. And granted, this is a little bit old, but she was looking at the number of papers that looked at primary production in systems, comparing pelagic, so that's water column, and benthic, or both. And so just to show how dominated the literature is in terms of actually what everyone looking at what's happening in phytoplankton, and saying there's almost nothing in primary production of the benthic zone, even though we can see here that that's a huge part of actually probably what we can expect to be happening in these systems. So it's a really fun place to be, because you can almost just have an idea and it's like, oh, no one's done that. No one's thinking about them. Something I also did right before moving here was I was actually working with a project looking at multiple stressors in the Great Lakes. So I was working at Guelph, just outside of Toronto. And the idea of multiple stressors is just taking in, there's a lot of people, for example, in the Great Lakes that are studying, just algal blooms in Lake Erie, or just zebra mussels, and just these invasive species, or just legacy contaminants. Multiple stressors, the theory is that all of these things are actually interacting with each other, and not enough people are trying to take this larger, broad image of seeing, okay, how do zebra mussels influence other invasive species that might be coming in, and do those feedback with the harmful algal blooms or hypoxic events, and is that possible that that can influence legacy chemicals? And maybe they'll cancel each other out, maybe they'll actually be sort of compounded issues. So again, here's a picture of Utah Lake, and I think this could be totally applied here, where you have invasive species and fish availability being two issues that people are maybe studying. Invasive species even being like Phragmites, for example. Food web interactions might interact with carbon cycling, legacy chemicals, ecosystem aesthetics, and then just trying to look at how all of these things interact. So I just kind of wanted to put this slide up there on the table as something that I'm interested in, and having this kind of broader idea. And I'm not just interested in benthic algae and what's happening at the bottom. I'm interested in how everything feeds back and how everything might communicate. So Peter kind of deflated this one a little bit by saying where I was, but I thought I would give a slide to show also where I've actually studied, keeping in mind what experiences I've had. So I did my master's in northern Quebec, so it's near James Bay, the bottom of Hudson's Bay. I was working on a reservoir there, a large hydroelectric reservoir, about 233 square miles surface area. And so that was sort of a boreal environment. That's me in the water looking at sediment respiration rates and looking at the contribution of the reservoir to CO2 emissions. After that, I did my PhD in Berlin, where I was studying two shallow lakes north of Berlin, a couple hours drive. So now down to 9 acres size, so pretty big shift and an average depth of 6 feet. And as you mentioned, I was looking at regime shifts there, and one lake, they had the same nutrient concentration, but one lake was clear water and one lake was turbid. And it was, so we were trying to see just the effect of these different communities. So the cleared water state was being maintained by a macrophyte community that was using some of the nutrients that were in there and keeping them from the phytoplankton. So you could kind of see, taking nutrients out of the equation, what the effect of the community was on how the lake processed carbon and what the production of the lake was. This then, as I mentioned now, is at the Great Lakes after that. So again, kind of jump up in scale to 31,700 square miles for Lake Superior. And there I was working on a larger project with multiple stressors, but also just trying to bring in some of this work I'd done on benthic algae further south. So there I am with, that was Yvonne Vadaboncoeur, who I've been working with out of Ohio. And again, we like to walk in the shallow areas and actually measure what's happening in these nearshore zones. And I'm going to go a little bit more into some of the findings I actually had, but this is just an overview of places. And then more recently, then I've also been up in a place called Nalidine Peninsula in Nunavut, so north shores of Hudson's Bay. working on a group of lakes that have that are all very shallow, benthic primary production dominated. It's part of a gold mine site that they're exploring, so they're doing an ecosystem assessment. And the great thing about this is I've gotten to a point where I can start sending students out. So that's kind of a nice progress I've seen from the first thing where I was the master's student out there at East Maine, and now I can have another master's student standing in the water in the Arctic taking samples for me. So starting with water quality, yeah, just to kind of keep track too, I've mentioned that I thought the most important issues were water quality, fish, and water quantity, so I'm going to track those on the top too. I already mentioned Utah Lake and Great Salt Lake as issues that seem to be important here and that I think are really fascinating. This was also something that came up. I was speaking to some people last week at USU, and this is an article from 2015. The headline says, "After Utah Lake related dog deaths, experts recommend protocol for toxic algae." So there's a lot of dogs that were dying when they were swimming during these harmful algal blooms in Utah Lake. So even at the very real social local level, it's an issue that people are worried about, and it's also an issue for fisheries, and it's obviously an issue for other reasons as well. Kind of separate to that, I've started, so a couple weeks ago I was meeting with some people from Bear Lake Watch, which is an NGO of the Bear Lake in the Idaho side, but they were interested in, for example, the sedimentation in Bear Lake, and they're setting up these monitoring buoys, buoys, which are...
We've been practicing that word.
Yeah, we practice buoy as a local thing. But yeah, so they're setting up a couple buoys to look at the long-term effects of what's going to be happening to the water quality in Bear Lake. But they weren't so much interested in eutrophication there as much as what sediments are coming in from the river, from Bear River, and how those are going to impact tourism. Also, there's a bit of a worry there. I understand the zebra mussels possibly invading. And some of the work I've done links a little bit with invasives I'll talk about later, but also this kind of loose tide brownification, and I'll talk about what I mean by that in a minute. But basically turbidity of systems, which actually might tie in as well with some of the sediment depositions we're getting associated with the wildfires recently here. So I mentioned this word regime shifts earlier, and I don't know how many people are too familiar with that, so I put together this cartoon. It's very basic, I'm not an artist. Just to explain what I mean by regime shifts and what I was doing in Germany. So this was a lot of my PhD work. And the idea for regime shifts is that you take a clear water, macrophyte-dominated lake, so something like this where you have nutrients going in from the system, and some of those nutrients are going to go to these algae these are the phytoplankton in the water column. A lot of them are also going to go to the benthic algae, that kind of green stripe along there, and those macrophytes. And so those can actually maintain this clear water state for a while as you're adding nutrients to it. So it doesn't instantly become turbid. It becomes turbid relatively slowly relative to the amount of nutrients you're adding. And also the macrophytes provide habitat, for example, for the zooplankton, which graze on the algae. So there's other controls which can come in which keep the algae in the water column down. But I mean, some phytoplankton are getting it, so it is getting a little bit more turbid each time you bring in more nutrients, and the nutrients are getting absorbed into the bottom. But as you keep on adding more nutrients to the system, you do get more and more turbid, and you can reach a critical turbidity where suddenly the macrophytes are no longer able to survive, and the benthic algae is no longer able to survive because they're shaded out by the phytoplankton in the water column. So you have this kind of switch, and the benthic algae are not really nutrient limited because they have a lot of nutrients usually in the sediments, but they're light limited. And then in the water column, the phytoplankton's nutrient limited, but not light limited. So there's this kind of feed trade-off between the two. And then you can, for example, the zooplankton go down because they can go down because you have more grazing of Nambai fish or more predation. So you can have these other food web effects, which really can suddenly change it into a turbid state. And you can call this a turbid phytoplankton-dominated state. And once you get there, it can actually be really hard to bring it back to a clear water state. And so this is what I was working on when we looked at regime shift. So it's not just eutrophication, but it's looking at how eutrophication interacts with the food web of the lake. And then you can actually have these other aspects. So once you, for example, lose that benthic algal community, you can have internal loading of nutrients as well, which can play a role. So that actually then stabilizes this phytoplankton dominance. If you have benthic anoxia, you get these chemical reactions at the sediment-water interface. So when the oxygen is gone down there, and the benthic algae are providing oxygen at that interface, once they're gone and they're shaded out, you can have nitrogen, phosphorus, carbon, dissolved organic carbon, iron, all of these things just flowing into the water internally from within the lake. So even if you're then cutting out what you're loading into the lake, you can have this really stable, hard-to-shake eutrophied ecosystem. And then and that kind of feeds back again to the phytoplankton. So it really raises this question, a lot of my work is looking what really is the importance of nutrients. So we really focus often on how much we're loading, but the idea behind it is that nutrients feed phytoplankton, zooplankton, phytoplankton feed the zooplankton feed the small fish and all that. But also, does a decline of one lead to a decline in the other? So if you cut nutrients, is this really all there is to the food web? And again, this ties in with what I was saying, how a lot of literature just focuses on the phytoplankton, which are nutrient limited, but ignore the benthic algae, which are not nutrient limited to begin with, but they're also part of the food web, and they're an important part of the food web. So there's more to it than just necessarily what's going in for nutrients. You have to also think about the light climate of these lakes to get an idea of where the productivity is occurring and how stable that is, maybe. If you have a lot of light, you're going to have more benthic algae, and that can be a control against this kind of internal nutrient loading, as well as providing fish food. So this was kind of the overview of what I was working at in Germany. We looked at, again, this turbid lake and a clear water lake. They had the same concentrations. And you don't need to look at that too much, but just to say that we found, in that case, for example, that the Clearwater Lake, despite having the same nutrient concentrations over the course of the year, was more productive than the turbid lake. So having access to light at the benthic zone, once you added in the benthic algae production and adding in there was also benthic cyanobacteria, there was macrophytes, and altogether they actually created more primary production over the course of a year than the turbid lake, which was mostly dominated just by algae in the water column, even having access to the same nutrients in the water column. So switching from that, I mentioned that I kind of teased about having brownification. So this is something that's a lot of people are talking about in Europe right now, where they're seeing, and in, I think more in, for example, around Ontario, Boreal, Canada, too. Lakes have been becoming darker, basically, over the last 20, 30 years. And there's a lot of different theories that are coming in. Some are saying that it's maybe related to changes in the acidity of the precipitation. If there's less acid rain, that can change the water chemistry. Others are associating it with climate change. For example, if there's more precipitation, you have more carbon being washed from the watershed into a lake. And it's probably one of these multiple stressor things, I'm imagining, where there's a lot of things that are happening at the same time and maybe influencing different lakes in different regions differently. But this is basically, when I talk about dissolved organic carbon, that's what makes the difference between, this leftmost lake water, for example, and then this one that looks like tea or coffee. And so often dissolved organic carbon, or DOC, has a lot of color, especially in the north. And that can have a pretty large impact then as well on, for example, benthic algae. If you're getting less and less light coming through, and if light is important for the functioning of a lake ecosystem, that can play a role in where the primary production is occurring in these systems. And this is particulate organic carbon in the deep brine layer of Great Salt Lake. So just to say that also a local example where you do have, again, changes in the color. For example, if you have part of the deep brine layer coming into Bear River Bay or these parts of the lake, where it might not in other times of the year be, that's going to be switching where your production is occurring. And so it's important to think about again, where the carbon is and where the shade is occurring in systems. And we saw this in in the turbid lake that we were working with in Germany, in one of those two lakes I was there 2010, 2011, and 2012 for my PhD. In 2010, these are 2010, this is 2011, and 2012, and we saw the water gauge level versus dissolved organic carbon rising. So what happened was in 2011, we actually had a record rainfall in Germany, and so the lake level started rising. And you can see as the lake level was rising, you had this slow increase in dissolved organic carbon. So it went up about 1 meter from where it had been. And all of a sudden, we saw this total shift where the lake level didn't really go up anymore, but in 2012, it suddenly went from roughly 25 milligrams carbon per liter to over 50. And actually going back, for example, to 2007 or some earlier data we had, you could see it used to always historically be around between 10 and 15 milligrams. So even in 2011, it was outside of what they had historically measured in this lake. And this is what the lake looks like from above. And what we realized was happening was that during this slow period where it was flooding, the lake was going into some of these sort of PD areas that stand around there. And the carbon from that area was leaching into the lake waters. So it was just this sort of passive diffusion that was going in there. However, here, so after 2012, we suddenly had the lake had become too turbid for benthic algae to be at the bottom zone, at the deepest zone. And so you could see that there was actually this internal release of carbon. What I was mentioning was like these internal nutrient loading. We also have internal carbon loading. And so we had nutrient data for all this period, and we were able to actually look at the fluorescence of the carbon to match these different sources to see that this was a different kind of carbon that was in the lake than this was during this period of rising. So we were able to kind of look at what we end up calling a brownification anoxia feedback, because once this started, once you have this internal loading, again, that shades out a larger area of the benthic zone. So it made the problem worse. It also was pumping nutrients into the system, so then you suddenly had more algae growing. And then once that happened, then you have, again, a larger area that's shaded out, and it went right to the edge. And so here's looking at the light availability during those 3 years as well. And this is depth of the photic zone divided by mean lake depth. So that's, for example, 1 is where the mean lake depth equals the amount of light, the light access up the sediment surface. So it means when you have more than 1, you're basically always having some light reaching the sediments at the mean lake depth. And you can see in 2010 it was generally always above that. In 2011 it was just touching that. And then in 2012, when we had that sudden crazy change in the system, was when suddenly the mean lake depth was outside of the photic zone. So light seemed to play a really important role in that sequence of events there. And this is just showing the same 3 years in a different way, and looking at the formation of anoxia in that system. And so again, this is kind of at the mean lake depth, is that black line, and this is the lake surface. You can see that there's anoxia below 1 milligrams per liter oxygen was only around the sediments in 2010. In 2011, it started coming up a little bit, a couple meters from the bottom, and then by 2012, we had it all the way to the surface, and it became almost a defining feature of the lake, and it was really stinky there, and there were too few macroinvertebrates or fish to even have population estimates to compare. So it was essentially a full fish kill in the lake. So looking forward, there's a couple projects that I would be working on here already that I've started organizing. One is looking at the paleolimnology of Utah Lake. So we're really interested in the history of eutrophication of the lake, and this would be with Janice Brainey, and who's a paleolimnologist at Utah State University. And also we're organizing it with Jeremy Gaeta, who's been working on Utah Lake for a while now. And so we'd be I mean, taking these sediment cores, you can go into the history of the lake and get some really cool data. For example, looking at the history of productivity, of algal productivity. You can see how much was benthic versus pelagic through the history of the lake in the past 150, 200 years. 150, yeah, 200 is a bit of a stretch. But you can look at the history of macrophytes. You can see if they're more submerged aquatic vegetation, which is a sign for the water clarity in the history of the lake. Diatoms, so those can again be pelagic or benthic. And so you get this idea of maybe a switch in the ecosystem from a clear water to a turbid state historically, and you could say if it's a well-dated core, what years or what decade that occurred in. You can also look at fishy DNA, so you could tie that theoretically with the introduction of carp or pike or these other introduced species where you know to the year that they were introduced. You can see what was happening in the ecosystem around that time. And also you can have an idea with the history of nutrient concentrations from these sediment cores, and that's something that Janice has been working on as a new sort of method to develop. And that can give us some idea also with nutrient loading. So all of these things together, it would be nice to kind of look back at the history of a lake like Utah Lake and see what really happened and piece together its recent history and what the role of people was. And also looking forward, as I mentioned, there are these problems with the buoys, or these possibilities of working with the buoys in Bear Lake. So that project that Bear Lake Watch has funding for now is funded for 2 years. And Dr. Brainy and I are planning on joining that with a course that would be doing annual monitoring of the lake and hopefully getting extended funding for more than 2 years so we can have long-term monitoring. And the value of that, I mean, Bear Lake is great right now, but again, there is this question of having baseline data and really understanding, you want to start studying a system before it's a problem system. So we have an idea of current water clarity, but before sediments become an issue, before zebra mussels are maybe introduced, before eutrophication possibly happens, you can have some kind of historical data set where you can see, okay, where did the problem come through? Looking at the mechanism of that problem and having enough data to really show what the mechanism was is the best way of figuring out how to resolve it. And if you just suddenly have a problem ecosystem where you don't have enough data to show you what really happened along the way, you can't, it's a lot harder to deal with. Another thing with water, is looking at avian botulism. So we've been contacted by some duck hunters at Great Salt Lake that are interested in possibly doing some project there, funding some projects there, which would be interesting to them. And I was actually out on Great Salt Lake at Bear River Bay yesterday and just seeing like a sea of dead birds, pintails. There's thousands of them. And I mean, that's also a water quality issue, and that's something that there's at least, Dr. Wurzbau has been looking at the possible relationship between cyanobacteria and harmful algal blooms and avian botulism in Great Salt Lake. So it's something I'm just starting to learn about myself. So moving forward to fish, just reiterating with the same graph that I had before that fish use food that's grown in the benthic environment. So that's so what I'm doing is totally applicable to understanding fish production and fish availability and the health of fish populations. And this is, for example, in Flaming Gorge Reservoir. This is a paper from Haddix and Phaedra, Tyler Haddix, looking at factors that limit growth and abundance of rainbow trout in Flaming Gorge. And they found, for example, that access to the benthic zone was essential for the success of the rainbow trout there. And so they were only really successful in the areas where they had that access. She can talk more about that later. But another thing that I've been working on is how, again, this relationship between algae and oxygen supply in the water column. So as I wrote here, algae can produce oxygen and take it away. And I'm going to give a really quick kind of look at the history of our understanding of how phytoplankton can produce hypoxia or anoxia systems. So this is Lake Erie, where I was working most recently. And the idea is that you have high nutrient loading, usually phosphorus is the target nutrient, and that produces a high phytoplankton production. in the water column. So you have a lot of algae being fed by these nutrients. Those settle to the hypolimnion and lead to large bacterial production in the hypolimnion, so in the bottom of the lake. Once you have all that phytoplankton, it's basically like a nice feast that's come down to the bottom of the lake to a place where there's not much light or no light. And that produces, this large bacterial production, and they use up the oxygen. So they're like us breathing out CO2 and breathing in oxygen. So what you call sediment oxygen demand increases. So they basically use up all of the oxygen at the bottom of the lake. And then if you have mixing, or depending on the extent of that, you can have this hypoxia, so just using up the oxygen. So if we look at the 1970s in Lake Erie, people were able to put this together and say, we can quantify the nutrient loading. We know that there's all this agriculture happening all around that lake. We can measure the phytoplankton production in the lake. We know it's massive. We know gravity was occurring. We know that phytoplankton increases bacterial production. They could measure the higher bacterial production. They could measure the sediment oxygen demand in the bottom and say, yes, this is extremely high. And then they were observing these fish kills in hypoxia. So that was the kind of thing where they were able to put this together and present to the government and say, we need to do something. And that's how the Great Lakes Water Quality Agreement 1972 ended up coming through, is they were able to show a really clear theoretical case and put it together with the data to get policy.
Is that another Canadian?
That was Trudeau. Yeah. So, and what we see in the history of Lake Erie's hypoxia is that there was a pretty good response to that change in policy. So this is hypoxic extent, 10,000 kilometers square. And this is starting in 1987, but they were showing that by 1996, you were basically almost down to zero. So they figured it was problem solved. You can see introduction of zebra mussels and reductions in nutrient loading were happening during that period. So the zebra mussels were also taking out phytoplankton from the water column, and so they were playing a role possibly as well. What happened after that, though, so 97 was actually the largest El Nino, I think, in history. I don't know if that had anything to do with it, but coinciding with that year, you suddenly have this return to hypoxic conditions. And kind of an interesting problem for me when I started doing my postdoc in Guelph, because you look through the history and you can see that total phosphorus had declined really dramatically in the lake. So this is from 1970 to 2010. Soluble reactive phosphorus might be a more complicated story, but it looks as though it either declined or stayed about the same. Also, water transparency declined. So this is looking at from 1970s to 2000s. In Lake Erie, and this is the Secchi depth. So in the central and west basin where most of the anoxia is occurring, you had Secchi depth readings that the water visibility declined by about 2 meters. And that's a lake that's, I think the mean depth of the west basin is about 7 meters, so it's pretty substantial compared to the depth of the lake. The center basin, I think, is 14 meters mean depth. Also, I don't show it here, but sediment oxygen demand declined, so you don't have as much bacterial production of the sediments that you used to. And so it looked as though all these indicators that we were using to diagnose the problem were changing. So looking at the 2000s now, you're able to kind of put together the same story. We say, yeah, we have hypoxia, but we don't have this, we've reduced our nutrient loading, we don't have this high phytoplankton production, sediment oxygen demand hasn't increased, it's gone down. And so it looks as though the mechanism behind this historical hypoxia is different from what's happening now with the modern hypoxia. So I'm just showing this quickly because we actually I sort of applied some of the thoughts behind this study in Germany, looking at brownification and anoxia feedback to see whether it's possible that there's also a role that light was playing in Lake Erie's hypoxia in the more recent years. Because it was also the only Great Lake in the last 40 years that's become more turbid. All the others became clearer. So we just published this in the Journal of Great Lakes Research, basically arguing that in around 1997, possibly associated with that El Nino year, there was a lot of high precipitation. There was a short-term spike in loading just from so much water coming into the system. And it's possible that that was kind of an analog condition to exacerbate the shading and shade out benthic algae. There's no data to show that there was a large internal loading of carbon in that kind of a feedback. But when you shade out benthic algae, you are actually making resuspension much more likely as an issue. And there was a lot of experimental support for that. So we were arguing kind of a similar mechanism, but at a different scale for a totally different system, and saying where benthic algae can actually stabilize. It forms a matrix that stabilizes the sediments. And so we're looking at the relationship now then at, is it possible that having this, this sort of perturbation created this return to conditions through a totally different mechanism, and we're having now basically a feedback loop that's supported not from phytoplankton production driving the anoxia, but from a lack of benthic algal production. And that decline in oxygen production at the base can actually have the same net effect as feeding the base of the food web. So one of the projects here in Utah that we've been talking about doing is actually, Phaedra and I were discussing on the way here, and this would be along with Jeremy Gata, linking benthic algal production to fish production, and even just, it's kind of a low-hanging fruit. This is from 1990. People have looked at this with chlorophyll and saying, so we know that fish are using benthic algae, but we don't know that, for example, if the system has more benthic algae, there's going to be more fish in that lake. Or are they just using whatever's there and it happens to be the best available food? So they showed in 1990, at least in these data set from Downing et al., that it seems in lakes that they had more chlorophyll in the water column, you had more fish biomass. And we were thinking it would be cool to put that together with, for example, even just lakes we have data on here in Utah, and seeing, is it the same relationship, or does it change if you actually start thinking about the whole lake food web? And it's kind of amazing that I haven't been able to find any study that already shows that, just seeing how important are benthic algae to fish in terms of their response to fish production. And then, you can start going into different questions here and saying, is this different between lakes or reservoirs in Utah? And there's lots of smaller questions you can start parsing out here, which I think would be really interesting. And I know I'm basically done, so maybe I'll just go forward very quickly through water quantity. The idea here that I had was that water quantity, drought in the west, you have different uses that are exacerbating water quantity availability, water availability in Utah. So our cultural practices, the politics determine how much we use and how much is used before, as it passes through rivers before reaching the terminal system. Then you have this issue of water supply, so whether climatic shifts or natural anthropogenic, long-term or short-term shifts in precipitation patterns are going to be affecting our water availability. And this is going into the carbon cycling part of my work, so I'm just going to go through it quickly because I think it's kind of interesting. But as I mentioned earlier, lakes generally pump CO2 into the atmosphere. So this is from 1994, looking at CO2 saturation of lakes in a large sampling. And they found that most of them, this is atmospheric saturation, so most of them are supersaturated with carbon dioxide. More recently, they tried to estimate the actual total petagrams carbon exported by lakes per year into the atmosphere. So that's 1.4, versus say around 30 for anthropogenic emissions. So they're still not emitting as much carbon dioxide as humans are, but we believe it's part of a natural regulation of carbon dioxide historically for the atmosphere. And it's also, and these numbers are very loose right now, and we're trying to figure out what kind of effects we have on the ecosystems that can change those numbers. And so this is one of the lakes I was studying in Germany. And you can use this kind of carbon cycling analysis to look at the lake history. So for example, looking at, so we would actually quantify just all the carbon that was going into the system. So looking at all the alder leaves, it was a closed lake with no surface inflows or outflows. Looking at the groundwater, we built about 10 or 11 wells around the side. And then you just try and match over the course of the year to see what was going in over 1 year and then what's coming out. So what's going into the sediments as permanent burial, and then it's totally removed from the atmosphere, and how much is going as CO2 into the atmosphere. So yeah, this tells you something about the lake history. When you get these paleo cores, you can start seeing how the lake has changed. We could see one of the lakes had become a turbid phytoplankton-dominated lake over a period of 30 to 40 years. It can also give us an idea of the storage of legacy contaminants. So a lake that has a very active benthic environment where there's lots of light, for example, might not bury loaded contaminants or pollutants as quickly as one where everything that hits the bottom of the benthic zone is buried instantly. And then again, you have this idea of how lakes might influence climate change. And if you have, for example, all the lakes in the tundra becoming more or less CO2, or loading more, or higher or lower sort of CO2 rates into the atmosphere, you can actually have theoretical large feedbacks or controls on anthropogenic climate change. So I'm going to leave it there. I had a few questions for you. So I mean, one of the questions is, I mean, this is going back to that first slide of, these different things that I thought were local issues, and also ways that local lakes might be changing. One question is even just, if anyone has any ideas about how the work that I've done would be useful to Utah in terms of managing Utah's lakes and reservoirs. And then these are kind of talking points that might go in there in terms of, again, looking at reservoirs versus lakes, looking at invasive species. If anyone has any special interests in or passions in that, looking at changing nutrient dynamics, or anything else that you guys think might be important to talk about. So thank you very much. Just time to go back to work now. Yeah.
Now, that one graph you had where you had basically the 57% of the contribution that came from the benthic proportion of the nutrients and whatnot.
Yeah.
There's a lot of scatter in those numbers. Any feel for how variation or how different variables of limnology affect the scatter on those numbers?
Yeah, so I imagine the scatter is largely linked, also the scatter here. So you can see, for example, some shallow lakes are 1 or 2 meters deep, and they can have anywhere from 20% benthic contribution to the whole lake carbon production to 80%. So you are going to have a lot of scatter. For example, a fish that's in a lake where 80% of what's in the lake, what's available as food, is in the water column, then that's going to become, it should theoretically become a larger part of the fish's diet. So again, yeah, this just kind of speaks to also what, on one end, it's kind of a question of optimist or pessimist. On one end, you can say it's amazing how much it can matter. Benthic carbon production can be in some of these fish about 100% of what they're eating, and in other lakes it's almost 0%. And so, to me, it's kind of a call to get to understand that process better. So these are just from stable isotope analyses from a large number of lakes. They could see that was there, and yeah, there's a huge scatter. And they suggested, for example, that it was that it looked as though it weighed more heavily than you would expect it to weigh based on the availability of benthic food. So, for example, there's another study I don't show here, but from 2014 in Lake Superior, and even there they found it's about 50% of Lake Superior's fish that they sampled of all of Lake Superior's fish, they sampled about 50% of the diet was benthic. So even in the largest, deepest of the Great Lakes, where they wouldn't have expected it to have a large role.
Those are direct part of the food chain, directly to the algae, as opposed to going through an insect or something like that.
So they've gone through the insect too,
Okay.
but they can see the insect ate something that was benthic. That was
Okay.
benthic algae. Yeah.
But before this paper came out, most traditional limnologists, and certainly almost all fish ecologists and biologists, completely ignored the bottom of lakes. So if it's 0 or 100%, it doesn't really matter. What matters is that there's a lot of it in some places, and it was always historically ignored.
Yeah, so one of the papers that I just published last year when I was working on the Great Lakes was just quantifying even theoretically how much it might be in the Great Lakes at the whole lake scale for each of the Great Lakes and how much it may have changed, say, with the zebra mussel introduction in the last 40 years. And, I mean, at the high end, using numbers that people had measured there, it looked as though, for example, Lake Huron might have 50% benthic pepper production, even though it's such a massively deep lake, but it's so clear water, and sometimes you have, visibility to 40 or 50 meters, and the lake's mean depth is 80 meters, and there's almost nothing growing in the water column. So you can start, piecing together the lake to get an idea of how important you would expect it to be to the fish, but then you need to start kind of going deeper into it to see whether those relationships actually exist.
Interesting, look at that data with eutrophication as kind of like a third axis.
Yeah.
Or axis, and looking at the effects of different species assemblages and things like that. It's kind of interesting data set, interesting to pick up more factors that affect the distribution.
Yeah, and that's Jake Van Der Zanden out of Wisconsin, and he's doing, I think he's still releasing, I don't know whether from this data set in particular, but he's doing a lot of really interesting work on this.
So you correlated the production or the benthic contribution with depth. Has anybody correlated with Sadecki depth?
With
So how clear the water is and what proportion of fishing?
yeah, so actually I have, I did that for the Great Lakes, the project I was working on. It's actually, we didn't do Secchi depth, but we did, we looked at sediment light availability. Because it was, so basically Secchi depth given the morphometry of the lake to see. In the Great Lakes over the last 40 years, we found that it was about, it was almost for the middle of the road estimates that we had, gave a 1-to-1 line. We had, for example, a year in which, or a decade in which Lake Erie, maybe only 5% of the sediments had light access. About 5% of the benthic, 5% of the total lake's primary production was benthic. And then when you have like years that were maybe up to 30% benthic light access, and it didn't seem to matter whether it was eutrophic or Clearwater Lake, you had 30% benthic proportion of the total food web. And that was, I think, kind of shocking for us. We were expecting Lake Erie to have tons of benthic primary production because it's so shallow, and Lake Superior to have almost nothing. But we found that it was compensated for by, the shallow lakes are generally more eutrophic and they have a lot more happening in the water column. So it actually sort of balanced out in terms of the relative importance of benthic production. It was almost higher in these clear, deep lakes than it was in these really shallow, turbid lakes. So, but yeah, I haven't seen anything like this specifically with water clarity, but that's something, hasn't been written yet, as far as I know.
From your experience or research, do you have any feel for how long it takes these regime shifts to take from a macrophyte driven to a phytoplankton?
It's a really interesting question. And we were actually still hoping to write a paper on regime shifts and even just people agreeing on what a regime shift is. Because it can be 1 year to the next, you'll see a lake seem to go from turbid to clear. People say, okay, that was a regime shift. With the lake we were looking at in Germany, we were looking at the sediment history, and it took about 40 years for the carbon cycling of that lake to change, where it kind of maxed out, and you could see the year it started, and then it took 40 years for the macrophyte community to totally disappear. And we were saying, okay, well, is the regime shift, should it just be something that's visible and you just see what's happening in the algae in the water column, top meter, or is it, is it better to think of them as this long process of shift in how the lake actually works and functions, almost like as a machine, but it's changing the way it works. And as far as I know, I've never seen any discussion of that even, and I tend to be more on maybe the conservative side in terms of thinking of regime shifts as a whole ecosystem change that can take a long period, it can take 30 or 40 years, whereas a lot of people just, will just think of it as being a 1 to 2 year thing, or even a seasonal thing.
So is it kind of an oscillation then, where it's flipping back and forth for a certain amount of time?
It can be. Yeah, and you do have some ecosystems that naturally do oscillate, and there's a lot of work even in Argentina on just these, I think it's like in the Pampas there, they have, maybe Pantanal rather. They have a lot of lakes that just every year, as far as long as people have known, they can switch between these different dominated states. So, yeah, it's almost, it depends what kind of ecosystem you're looking at. And there's even hypothetical aspects where people say maybe really large ecosystems like oceans can undergo regime shifts, but they're just so huge that it takes them, like a century to do what a small, shallow lake would do in a year. Maybe they're just happening so slowly that we aren't really recognizing it. But it's still kind of an open, new theory in terms of way of understanding how lakes work. Yeah.
You dealt a lot with natural lakes in your research, and we don't have many natural lakes here in Utah. Most of our systems are impoundments.
Mm-hmm.
And the regime shift theory, if it is now a theory, for me, is very applicable to reservoirs because of the aging of reservoirs, and we are indeed seeing a shift in how those systems function. Regime shift, I would guess, then would cascade all the way up through the trophic levels and affect your fish community and how you manage those fisheries. Is there, to your knowledge, anything out there that describes how one might go about reversing a regime shift?
There's a lot of work on that now in Europe, actually. I haven't seen it so much there, looking at reservoirs in particular, but I think the theory and the mechanisms would be the same between aquatic systems. You have to, once you have an understanding of the role of different producers playing in light availability and nutrients, then it should be pretty directly applicable. They have reversed regime shifts there in lakes that they've been trying to, and often it involves, either fish removal if there's a fish that's making it more turbid. But again, this gets back to the point of, if you remove a bunch of fish and then it becomes clear the next year, have you actually reversed the regime shift, or you just made it clear for a year? And maybe you need to wait 20, 30 years for the whole macrophyte community to build up again, and to have this sort of stable base, keeping the sediments from resuspending and keeping these internal nutrient loading events from happening. And then you could possibly reintroduce the fish, and they won't have that same kind of devastating impact on it. So I think it's something that is kind of nuanced, and you'd have to deal case by case and looking at the lake. But theoretically, yeah, it's possible to reverse it.
The nice thing about reservoirs is you have another knob to turn because you can control how much water is in there.
Mm-hmm. Which controls how much sediment light access there is.
Yeah.
We've enjoyed great success knocking down carp in Utah Lake. Water quality's improved. Getting these harmful algal blooms, presumably benthic cyanos. What's a manager to do? One dial that's apparently untouchable is phosphorus loading. Then there's other dials we could play with, like reestablishing floating macrophytes.
What about that? Specifically floating macrophytes, or just Utah Lake in general? Yeah.
There's endangered species to recover. There's lots of people live there. Dogs dying. If you can kill a dog, you could kill a person. I mean,
Yeah. I mean, I think so. I was speaking a little bit with Jeremy Gator, been working on this. From my understanding, there hasn't been too much work on internal nutrient loading in Utah Lake yet. And I mean, that's where I would start, at least understanding, how much of that could be part of the problem versus just what we're loading from the outside. Because if you have that, then you can start piecing together, okay, what's causing the internal nutrient loading? And, is this a role that, macrophytes could be one thing in terms of keeping the water clarity, but they would also reduce that. So knowing that to me seems, kind of as an outsider coming into it, the first place I would start looking at how to fix Utah Lake. And I don't think it would be that easy, but at least knowing where the problem is the best way to addressing the problem.
And I guess, yeah, help us pose the questions.
Yeah.
I guess.
know that some people have to go. I think we should, this is a wonderful dialogue, but I think we should thank Soren again, and if you want to stay and ask questions, we're not in a hurry.